Good morning and welcome to this webcast. We're glad you're joining us here for this special presentation by STEMnet at the Office of the Chancellor. I'm Jennifer Wicks from Learning and Development. Happy to join you as your host behind the scenes this morning, and appreciate your interaction and your comments and questions in that chat pod on the left of the screen throughout the session today, and look forward to the Q&A. Really at the end of the session we have a full 2 hours of incredible presentations with our exemplars of engineering in the CSU system today, so you're really in for a treat. I'd like to introduce to you Dr. Frank A. Gomez. He is our Executive Director of STEMnet at the Office of the Chancellor. And with that, I'll go ahead and hand it over to you, Dr. Gomez, to begin.
Jennifer, thank you for the kind introduction, and welcome to all of you to another STEMnet webcast. I hope you and all your families are healthy and safe. Today's webcast is the first in occasional disciplinary-based webcasts highlighting exemplary research in the CSU system. Our theme today is Exemplars in Engineering. First, some numbers. In a typical year, California awards nearly 12 and 16% of the nation's bachelor's and master's degrees in engineering respectively. Of those percentages, the CSU awards over 51 and 31% of the state's BS and MS degrees. Quite an accomplishment. These numbers reflect the impact the CSU has in building the engineering workforce of California. Probably like many of you, I've asked students why they have chosen to major in engineering. Some of the reasons they have given are employability, high income potential, and ability to work internationally. But more and more what resonates with students is their desire to improve the world, to contribute to society. There is an increased yearning to take on today's current challenges and to solve everyday problems to improve our way of life. In the CSU, more so than any other system in the country, we provide research opportunities for students, the majority undergraduate, to apply what they learn in the classroom. It is by examining real-world problems that they gain the experience to compete in the real world. May this be in pursuing graduate degrees, entering the industrial or governmental sector, or private entrepreneurship. It gives me great pleasure to bring together CSU exemplars in engineering, reflecting some of the very best from across the CSU. Today you will hear a small sampling of exciting interdisciplinary research that provides our students with a great examples of some of the great work that we are doing here in the CSU, and that provides them a great experiential research experience. Before we begin, I want to remind the audience to use the open chat box on their screens to type in their questions. Our speakers, when not responding, will make every effort to respond in real time. So let's begin. I look forward to hearing from our presenters. Our first speaker is Chris Bachman from Cal State LA. Chris, the floor is yours.
Thank you, Frank. So my name is Chris Bachman. I'm the PI for the Sustainable Technology Lab at Cal State Los Angeles in the Mechanical Engineering Department. And I'm going to talk a little bit about some of the electrochemical energy storage projects they're working on for sustainable technologies. So our group is really excited and interested in electrochemical energy storage for its ability to promote novel and clean technologies. I think most people in the room, being from California, will be aware of the duck curve associated with the large use of solar in the state on our power grid, and the need for some energy storage for that problem, but as well as the ability for energy storage to promote technologies like electric vehicles or new portable applications, anything from biomedical implants or devices, from the drastic growth of sensors to military applications. So today I'm going to talk about two of the projects that we're working on in our group. The first will focus a little bit more on the transportation and portable sectors with the computational project that we're working on, and then I'll talk a little bit about a project more in kind of the renewable space that we're doing a little bit more experimental and analytical work on. So what's kind of dominating the supply for all of these applications right now are lithium-ion batteries. And the basic structure of one of these lithium lithium-ion batteries is that they'll have a negative electrode and a positive electrode. And they'll just be made of particles that are kind of glued together with a binder and separated by a piece of porous plastic. And the whole thing gets infiltrated with this liquid electrolyte, is what we call it. And the purpose of that liquid electrolyte is just to allow lithium ions to travel from the graphite that's being used in the negative electrode to this lithium metal oxide that's in the positive electrode, but to block any electrons. And the electrons get forced around the circuit, and that's where we harness the energy associated with the chemical reactions in the battery. But there are some downsides with doing it in this manner. This liquid electrolyte is typically an organic liquid, so it'll be flammable. And so when you have these battery fires, most of the energy released in these battery fires is associated with the energy of combustion of this somewhat inactive material in your cell. The other downside with this current technology is that you are forced to use graphite instead of lithium metal, which offers the promise to have large increases in the energy densities of the cell. So kind of a novel idea that's been worked on for a little while now is to replace this liquid with a solid material that serves the same function. And that will, one, it's a little bit safer because these materials are generally non-flammable, and you can use this lithium metal. So this will allow you to have drastic reductions in the volume and mass. The liquid electrolyte can also decompose over time, so this can improve the life and the safety of the cell. But there are some limitations to why we aren't doing this right now, and why this is not the current technology that's being used for these applications. And one is some of these newer materials, they're not completely stable with all the other materials in the system. It's harder to have a solid material that's as conductive as a liquid. And when we go to make the real cells and not the lab scale cells, we have trouble with processing and controlling the microstructure of these solid state electrolytes, is what they call them. So some of our work in this is we've partnered with Draper Labs in Boston, Massachusetts. And there's a group there that designs and builds the physical solid state batteries. But some of their issues is they need their experimental landscape to be narrowed so they can know what sort of materials should we be looking at, what sort of sizes should we be using in the cell. And so we partner with them and do the computational side of it. So we, in essence, we just track the lithium ions going through the cell. And we use that using kind of a common diffusion equation that's modified to account for things like the electric field is pushing the lithium ions around, and you have the ions can merge together and go away, and so you have kind of just a modified diffusion equation where we track the motion of the lithium ions in the cell. And so this is, for example, the concentration of lithium ions in the electrolyte while the cell is discharging. So if you look on kind of the far left, it's kind of flat, and then you see as you kind of come to the right, things tend to diverge, and you have high concentration of lithium near the negative electrode, and it gets lower near the positive electrode where it's kind of being sucked in. And the beauty of doing this in the electrolyte and the electrodes is you can begin to predict the cell performance very quickly in a lot less time than it would take you to make this cell and run the measurements on it. And not only can you get the performance of the cell, which is typically characterized like by a voltage versus time curve, but you can also find the losses associated in the cell with the different with their different causes, which allows you to kind of identify, all right, in this one case it looks pretty good, but maybe the charge transfer losses are very high, so we're going to need to do something to modify that. And so this really allows you to narrow down the landscape and allow them to build much fewer cells around kind of an optimized performance. And one thing that has become kind of now that for this technology, it's getting closer and closer to something that could be commercialized, you start running into more more kind of engineering problems associated with just making the cells. And so on the left, this is a plot of the voltage in the solid state electrolyte just over a grid. And so you can not only see that there's kind of a gradient up and down, but there's kind of a slight mesh behind it. And that's because in the real solid state materials, you have things like grain boundaries, you have things like voids, and those are imperfect materials. And so being able to model this mixture of voids, grain boundaries, these three phases in the system is rather important. And so we use a network of resistors to model this and be able to predict the total conductance of our solid state electrolytes. So here's like an example of the total conductance as a function of two different phases when the phases have different conductivities. So you can then begin to see, all right, so when the experimentalists make this solid state electrolyte, and what the microstructures are, you can begin to say, okay, this is what the real cell performance is going to be under these ranges of voids, and begin to make criteria for what are acceptable percentages of voids, what are acceptable sizes for the grains, thickness of the grain boundaries, and really allows this experimentalist to go through at a much faster pace. So that's a little bit of the kind of one of our kind of newer projects that we've been working on, associated with computational work more in the solid state electrolytes, because energy density is such a benefit. You really see that in the portable and transportation areas. Another kind of a side, another side of the group, we work more on kind of this renewable landscape. And so in that area, even though right now it's being dominated by lithium-ion batteries, it's likely this will get replaced with something like a flow battery. And so the difference between a typical battery and a flow battery is that the flow battery, instead of having solid electrodes, you actually just dissolve your active materials in these large tanks. And then you flow those into a cell that reacts. So if you're familiar with fuel cells, it's similar to that, except for the reversible. And the beauty of this is that, if you need more energy storage, you don't need a bunch of little cells all stacked together. You just increase the size of the tank, add more active material into it, and you'll have higher energy. And the additional benefit is that the size of your tank is not related to the size where the chemical reactions happen, which is decouples. You can design a system that has different energy density and power density to whatever you want, which is not possible in most typical batteries. So some of the newer ideas in this space, or most of the work in this space is done where the electrolyte is an acid, which is not ideal for large scale. We're talking about grid scale energy projects to have this corrosive, environmentally unfriendly kind of material at huge scales. So some of our work is can we do this with a neutral pH electrolyte? Can we dissolve our active materials in something like water and salt, basically? And so one, it's much less expensive, and it's non-toxic, and it's non-corrosive. So some of the work in my group has been to go through and catalog all the different materials that are out there in similar systems, and begin to say, what are some of these materials that would have potential in this system? And so here's a plot of kind of the energy density possibility of materials that have been used in relative systems against their voltage. And so the energy density in these flow batteries is really about, so up in the top right corner, the F and the N are constants, and then you have concentration, C, and voltage. And so it's really about you want something where you can dissolve a lot of it in the tanks, because that'll increase how much energy is in the tank. And you want to have a large voltage difference. But these these kind of highlighted black lines at the edge, is you can only pick materials with a certain voltage far enough away, because as most people are aware of, you can split water electrically if your voltage is large enough. And so so you're limited by this. So you want the highest energy density possible, but you got to stick in this window, otherwise you're beginning to create hydrogen and oxygen in your system, which is unwanted. And so that's for some of the reason why there's also proposed systems where you go to an organic liquid, where this window gets to be 4 volts or something much larger. So my students did kind of a thorough review of the materials, picked out some of the kind of highest potential materials in this landscape, and we're now kind of at the process to begin to screen some of these materials in this actual system. So what we do at this point is we take it into a 3-electrode cell. So and we run what's called the cyclic voltammetry. You sweep the voltage and measure the current. And so here's kind of just a representative plot of the black is kind of a black line or a blank in the system, kind of a baseline, and then 2 different iron-based compounds where you can see kind of drastic difference in the performance, where the red is kind of an ideal compound that we like for, or has high potential for being used in the system. And blue, you get this kind of, it's a little bit wider, which I don't want to go too much into electrochemistry, but shows it has a very different response than we would expect, and would kind of say like, well, this one's maybe one to steer away from, and the red one would be good. And so at this point, we're kind of screened a lot of the materials that are out there, identified good positive and negative materials, and then begin to test some of these new materials in this kind of system in kind of a lab scale flow battery to begin to optimize some of that cell performance. So with that, that's just kind of a little bit of some of the work done in my group surrounding energy storage. I'd like to thank kind of some of the amazing group members that have done some of this work. So Maya Hori and Blue on the left, she's the one who's doing most of the computational work in the group, and then Allison and Asher, 2 individuals on the right, they're the ones doing some of the experimental work. And so with that, I'll turn it over to my colleague, Perla Ayala, and let her take it from here.
Thank you, Chris. Good morning, everybody. My name is Perla Ayala. I'm a professor at California State University, Long Beach, and I'm excited to be able to share some of our work with you today. The title today is Engineering Models for Tissue Repair, and I will explain what that means for our lab. My area of research is tissue engineering, and so with this field, we try to engineer therapies that can help regenerate, repair, or replace tissues, and we do this by integrating different components from materials that are natural, biological, synthetic, using cells that can be autologous, they can be from other donors, and drugs and factors, and doing the combination of this to create the best therapy for the patient. And so just to kind of get this, set the setting for what we want to do, a little review about the repair process just in general. Of course, it will vary from tissue to tissue, but if there's some injury, some pathological disease, there might be some inflammation that occurs. And if the tissue has some capacity for regeneration, this will happen. But what happens is that in many cases, what dominates is this remodeling process where there's not enough regeneration, but there's still some kind of repair, and that becomes a scar tissue. This can go into pathological fibrosis, which will be an overproduction of the extracellular matrix as a response to this tissue damage. And so we're trying to find this balance of regeneration that we can get some functional tissue. That's kind of the goal that we're working towards. And so in particular, for my lab, we are focused on translating mechanisms of tissue repair into feasible therapies that will promote optimal healing. So with this, we have a couple of objectives. We have 2 projects that I'll present today. One is focused on designing a scalable, implantable bioscaffold for tissue repair and regeneration, but also developing functional 3D models that can help us understand disease and also do some screening of potential therapeutics through these models. One goal that we're trying to address, or one challenge that we're trying to address, is volumetric muscle loss, which is severe muscle damage that can occur with trauma. And if it's significant enough, it usually results in fibrosis at that site, scar tissue formation. So we know the muscle has some capacity for regeneration, and actually very good capacity for regeneration. But when the damage is significant, the only way to repair it is through scar tissue formation. So as you can imagine, that can lead to dysfunction in the tissue that can result in long-term disability. And so it can be pretty costly in terms of healthcare costs with that. And so we have this goal to design a biomimetic implantable scaffold to increase and direct skeletal muscle regeneration. So there are some challenges with creating these scaffolds. It's a significant amount of tissue that you would have to develop. It has to be scalable. It has to be mechanically robust for this type of application. And so those are some of the challenges that we have in terms of creating scaffolds. And so we're also looking for a tissue that will incorporate into the host tissues, a scaffold that will incorporate. And so we're looking at collagen. Collagen is one of the main proteins, structural proteins in the body, and if we can fashion it into a mechanically stable, robust tissue that can allow for muscle growth, it could be a significant advance. And so in general, collagen has been used for different repair applications, but it's usually presented in the form of a gel or a hydrogel, which doesn't have the mechanical properties for some of these other type of applications in terms of an implantable scaffold. So we're fashioning this collagen into sheets that could be then be stackable and create stronger constructs. In addition to that, we can improve integration, vascularization by adding some patterns, in this case porosity, that will allow for integration with the host tissue, and vascularization that can help with that integration process. But we're also looking at ways that we can improve muscle growth, so tissue growth within the scaffolds. And that's the project that we're currently working on in my lab with some of my current students. And so when we think about creating scaffolds for muscle repair, we can just say, well, you can just create a scaffold, allow it to integrate with tissue. But if we just have a scaffold with no significant guidance for the cells, we might just get incomplete repair. What we're trying to do is to really provide unique guidance and signals to the muscle scaffold that will allow for this complete muscle repair once it's implanted. And so what we're doing in my lab is we're able to extract and purify collagen from different animal tissues, and then we can create these collagen sheets. In addition to that, we want to add some guidance for the cells to grow on these collagen sheets. And so we created a PDMS mold with some microgroups where we can develop these sheets that would then have these microstructures on the surface that will allow for directed cell growth of the muscle cells, this myoblast that I will show you in a moment. And so what we've been able to do is to show how organized we can create this muscle tissue. We just look at a flat tissue in the first figure. A flat collagen sheet will not generate a nice organized tissue, but if we provide this alignment for the cells, they can start to create this more organized muscle tissue. And we've been able to culture these scaffolds for about 21 days, where we start to see more maturation of the muscle tissue. We're also able to show that we can increase maturation by this expression of alpha-actinin specific to muscle tissue, which gets enhanced once the cells are grown in this scaffold with the microgrooves compared to just scaffolds that are flat. Additionally, as I mentioned, we're trying to provide the best signals for this to happen. And so one way that we can do this is to try to incorporate the signals that the muscle cells see in their own environment. So muscle proteins, extracellular matrix from their microenvironment. So we are decellularizing muscle tissue and creating this powder of muscle tissue that can be incorporated with our scaffolds. And so our preliminary work shows that we incorporate these proteins that are directly derived from muscle tissue. We see an increase in myoblast proliferation, which it's really exciting to see that these signals really can enhance the proliferation and the maturation of muscle tissue. So this is kind of where we are. We have other results, but just in a matter of time, I'll be happy to talk to you separate on that. I also like to show you another project, and this is related to fibrosis after a heart attack. Significant fibrosis after a heart attack can lead to heart failure, so it's a very significant challenge in the clinic right now to treat patients. And so we want to establish an engineered model of cardiac fibrosis to try to identify critical players and signals that can help later to provide some therapeutic for this extra production of extracellular matrix after heart attack. And so we use this PDMS mold again to create the scaffolds that, in this case, we're trying to direct cardiac cells to be organized. As you know, cardiac cells also need to be organized, just it's another type of muscle to function correctly. And so this allows us to create a more accurate representation of cardiac tissue in the laboratory. In addition to that, we're also looking at the fibroblasts, which are the cells that are producing the scar tissue formation. So we're able to create models of normal versus fibrotic conditions. We can start analyzing fibroblasts and maybe analyze different therapeutics that can target this type of cells. So in this case, we're looking at the extracellular density, how extracellular density affects, extracellular matrix density affects fibroblasts. So here we have 2 milligrams versus 4 milligrams per mil, and we see an increase in proliferation once we have this increased density, which also results in higher stiffness for the tissue. And so we're looking at these two concepts of implantable scaffolds, models for fibrotic conditions, and so we're looking forward to continue to expand on this. My lab is consists mainly of undergraduate students and a couple of graduate students. Actually, many of my undergraduates graduated this spring, and a few are graduating in the fall, but they've been able to advance this work quite a bit, and I'm also very grateful for the funding that has helped support this research. And with that, I would like to thank you for the opportunity, and I'd like to introduce our next speaker, Sankha Banerjee from State Fresno.
Thank you, Dr. Ayala. Good morning, everyone. I'm Sankha Banerjee. I am the PI of the Energy Devices and Plasma Applications Lab at Fresno State. Today, I'm going to talk about the design and fabrication of electro and photoactive materials for applications in biomedical devices and water purification. Our lab deals with the full life cycle of these electro and photoactive materials. So we start from synthesis and fabrication of these materials, and eventually we go into processing where we look into both chemical processing and plasma processing. And eventually, we also look into the performance of these materials. So the goal of this lab is to develop the interconnectivity and relationships between fabrication and synthesis, materials processing, and its eventual performance. So today, with the interest in time, I'm going to discuss about 3 major areas that we work in the lab. The first area is looking into materials fabrication, processing, and characterization. And the second area would be the application of these fabricated and synthesized materials, which are processed using chemical methods and plasma and electrical plasma application methods in wastewater purification, desalination, and also application in biomedical devices. We also work in other applications as well. We look into applications of these materials as sensors or active sensors, in civil engineering, and also in many other devices. But today, I'm just going to talk about these 2 major application areas. Now, in our lab, we start with synthesizing nanomaterials such as zinc oxide, barium titanate, or even graphene-based materials. And eventually, we look into fabrication methods to develop bulk materials and thin films, which can be eventually used either as sensors or energy conversion devices in either biomedical devices, civil engineering applications, or in water purification. So in the images that you see right here, I put a few microstructures. These are all scanning electron micrograph images. We have one EDX image as well. We deposit zinc oxide-based composite materials. In this case, the image that you're seeing right here is zinc oxide, an epoxy flexible material that is deposited on a stainless steel flexible substrate. We also deposit materials on transparent polymer substrates, And these materials are then processed using chemical methods. So we look into bulk materials processing. We also look into surface modification of these materials. Now, it is really important to understand the microstructure of these materials and how the different phases in these materials interact with each other. So we look into the microstructure using scanning electron microscopy and transmission electron microscopy, and we try to understand different aspects like agglomeration, also looking into percolation, so that we can understand the different mechanisms for electron transport. So in this case, in this specific example, we're looking at a zinc oxide epoxy composite, and we can also add a conductor material such as graphene or carbon nanotubes that is going to tailor the electron transport properties from the bottom of the thin film to the top of the thin film. And based on the tuning of this electron transport properties, we are able to get our desired performance for specific applications. Now, the next area we work on, as I mentioned before, is looking into plasma processing of these materials. So we are looking into, initially we started off with looking into chemical processing, and we also did assisted plasma processing in addition to chemical processing. But we are trying to transition to complete plasma processing, because if we want to apply these materials in biomedical wearable devices or implantable devices, then we do not want any kind of toxic residue or toxic interactions with living cells, so or with our skin. So in that case, we are trying to move into plasma processing. We mostly use room temperature plasma to process this material. So the images that you see here are corona discharge based plasma processing or surface modification of these materials. What we're trying to do is trying to modify the surface energy of these materials. Now, if we modify the surface energy, these materials might have better bonding with the electrode or better bonding with other thin films that are stacked on top, or even better bonding with the device that these materials are attached to. We also look into the hydrophilic and hydrophobic nature of these materials, and that might also help us in developing scaffold and also growing specific kind of cells, and also inhibit the growth of, let's say, virus proteins. The next area I'm going to talk about is water purification and desalination based on the materials that we synthesize in the lab or fabricate in the lab. The first project here that I will talk about is a project that we started in around 2016. In this project, we looked into developing water filtration stacks or water purification systems with graphene monolayers. There were a lot of issues in this project with related to fouling and clogging of the different nanopores in the graphene microstructure. We also tried to control the defects in the graphene monolayers. So by controlling defects, we were able to selectively remove certain ions or certain impurities in wastewater. Now, moving on from this project, we were trying to commercialize this idea, and eventually we moved into looking into existing systems. So some of the existing systems that are available in the market mostly work on activated carbon. So we looked into how to couple this activated carbon-based water purification systems with surface modified or doped graphene-based materials. And eventually we looked into graphene nanoplatelets. So we developed filter stacks in combination with the existing systems, and we looked at the performance between graphene and also the activated carbon-based filter stacks. So graphene-based filter stacks gave us a better performance, and the TDS, that is the total dissolved solids in water, and the conductivity, those were controlled after only 3 cycles, as compared to activated carbon, where we could go down to drinking water after 4 cycles. So in combination with activated carbon, this system was more efficient than only using either graphene or activated carbon systems. So we're looking more into this, and we're also trying to add an aspect of using capacitive deionization, where we will try to control which specific ion we want to remove from the wastewater. We are also trying to couple this with a pre-processing using a plasma discharge system, and we have developed we have developed a system in the lab which looks into applying microplasma and trying to purify water in situ. So if you look into the bottom left figure, you would see that we are trying to apply microplasma using hypodermic needles, where we shoot the water out of those needles as a water mist. And this water mist is combined with the room temperature plasma, and that helps us in pre-processing and removal of any kind of organic contaminants in water. So this system, coupled with the previous system, can actually help us removing both organics and also specific salt ions or other kind of ions that are actually impurities in water. The next area of application is looking into biomedical devices based on the electro and photoactive materials that we fabricate in the lab. We look into a lot of photoactive material fabrication as well. So back in 2016, we started looking into fabrication of perovskite-based photoactive materials for the applications in solar cells. Now we looked into non-toxic materials such as bismuth and tin, and we found out that these materials can also be applied in biomedical devices as photoactive sensors. So we work on both the experimental and simulation side, and currently we are looking into bismuth and tin-based materials for application as photoactive sensors in biomedical wearable devices. Now the next project is actually based on this and also plasma processing. So we are trying to develop systems in addition to processing methods using room temperature plasma. We were trying to modify the surfaces of biocompatible composite materials so that we can develop scaffolds that can help in cell selective growth. We are also trying to use the photoactive materials that were described in the previous project to try to develop biomedical wearable devices for medical diagnostics, such as continuous non-invasive glucose monitoring, and also eventually looking into other health metrics as well. One of the other projects I would mention quickly with interest in time is the robotic arm-based material handling project. So this was a senior design project that we have been working in the past 2 years with the goal in mind, so that we can develop AI machines to run automated experiments inside the fume hood. So in this case, the robotic arm is programmed to run specific experiments, and we are also trying to develop machine vision-based algorithms and also looking into artificial intelligence algorithms so that these robotic arms can autonomously run these experiments rather than researchers running these kind of experiments. This will be really helpful for us in the future. I'm working on this project with my collaborators at Fresno State and also at SUNY Buffalo. If you want to check out the other projects in the lab, you can visit the website www.energyandplasma.com. And if you want to get in touch with me, I'm definitely looking for collaborators in the area of surface modification of biocompatible materials using room temperature plasma for application in cell selective growth. Before ending this presentation, I would like to thank all my lab alumni who without whose help we couldn't reach at this level and compete nationally. And we have published a bunch of papers with them, so I would like to thank them. I would like to thank the current group members in the lab, the graduate students and the undergraduate students, my collaborators at UC Merced, UCSF, Fresno, Buffalo State, and the Princeton Plaza Physics Lab, and also the different funding agencies. With this, I would like to introduce Dr. Christy Dykstra from San Diego State University. She is going to present on resource recovery using biochemical systems.
Thank you. So I'm an assistant professor at San Diego State University, and I focus on energy and nutrient recovery from wastewater treatment. And one of the ways that we do that in our lab is using bioelectrochemical systems. So I'd like to first start off with a little bit of a background on what bioelectrochemical systems are. So like a chemical fuel cell or battery, it has an anode and a cathode that's connected through a circuit, and an optional proton exchange membrane. The anode and cathode compartments have electrolytes in them. However, in a bioelectrochemical system, we use microorganisms as self-renewing catalysts to catalyze the oxidation and reduction reactions in the anode and cathode, respectively. So for an energy recovery bioelectrochemical system, we can use wastewater as a feed source for the microorganisms in a bioanode. They can oxidize those organics and release electrons to the solid surface. Those electrons can travel through the circuit, and by applying a very small voltage in the cathode, a different set of microorganisms in the biocathode can take those electrons and take protons released from the anode and convert CO2 to methane. So in this way, we can recycle CO2 into methane for a carbon neutral fuel. So how would these work within a wastewater treatment system? Here we can see a process diagram of a typical wastewater treatment system. And most of our wastewater is treated through an activated sludge system, which is a very expensive process, often the most expensive process in wastewater treatment due to the energy requirements for aeration. Some energy can be recovered through anaerobic digestion, which produces a biogas that has a mixture of CO2, methane, and other trace gases. However, because of the high CO2 content in this biogas, in order to use it, biogas upgrading must be conducted or expensive combined heat and power equipment must be utilized. And in the case of typical biogas upgrading, the CO2 is separated from the methane, and the CO2 is now a waste product. However, with a bioelectrochemical system, we can take a stream of wastewater prior to the activated sludge, treat that in the anode, so we can oxidize that in the bioanode without aeration, and this saves us energy costs by reducing the aeration requirement for activated sludge. Simultaneously, in the biocathode, we can take the biogas from anaerobic digestion, and instead of separating out the CO2 from the methane, we can directly convert the CO2 into methane, creating natural gas quality biogas that can either be used on site or can be injected back into a natural gas pipeline. When we think about biogas or energy and nutrient recovery from bioelectrochemical systems, we can look at three different areas: energy, nutrients, and water. So the first areas that we have focused on have been energy and water treatment. So for the energy, we've looked at the role of iron in a biocathode, how biogas trace gases affect performance in a biocathode, and now we are actually pioneering the application of synthetic biology to improving energy recovery. In the bioanode side, we can improve wastewater treatment by improving reactor architecture, evaluating how the wastewater feedstocks affect performance, and also using our synthetic biology tools to improve wastewater oxidation and organic removal. We're also looking to include nutrient recovery in these bioelectrochemical systems. And right now we're currently working on the proof of concept of a system that will integrate both wastewater treatment, energy recovery, and nutrient recovery in the form of struvite precipitation. And furthermore, our group is also producing or working on a model that will be the first comprehensive model of a bioelectrochemical system that includes a bioanode, biocathode, and gas transport between the two compartments. So a couple of the activities that I wanted to highlight today is, first of all, we have developed a zero-valent iron amended biocathode. And the hypothesis behind this was that if we take metallic iron or zero-valent iron and we put it in the biocathode under reducing conditions, it produces ferrous ions and hydrogen. And that hydrogen can be used as an electron source for the reduction of CO2 to methane. And indeed, the microorganisms that do this in the biocathode are very happy to use the hydrogen and improve the methane output using a simple addition of iron. The second project that I wanted to highlight is our evaluation of how hydrogen sulfide as a trace gas affects the biocathode performance. So hydrogen sulfide is a common trace gas in anaerobic digestion, and depending on the sulfur content of the wastewater going into the anaerobic digester, you will have varying amounts of hydrogen sulfide produced in the biogas. It's a toxic, corrosive gas, it's undesirable, it causes odor problems, and has to be removed prior to combustion of biogas. It's also often toxic to microorganisms, so we wanted to look at how that hydrogen sulfide in that biogas would affect the biocathode performance. So first, what we found with our zero-valent iron amended biocathode, you can see on the left in this plot, our red and blue lines represent our zero-valent iron amended biocathodes at 1 and 2 grams per liter, and the black line represents our control biocathode. At day 0, or time 0, we added the iron to the amended biocathodes, and each vertical line that you see is a time when we completely switched out the medium, and we did not add any more zero-valent iron. Yet, despite that, we see elevated amounts of methane production throughout over 4 feeding cycles without any more addition of iron. So we decided to look into this a little bit more to better understand it, and it seems that there may be a role for a redox active precipitate that forms and coats the biocathode. And you can see on the pictures that it is white when it's in a reducing environment. When it contacts oxygen, it turns blue, and it can be electrochemically oxidized even further to a yellow-colored substance. We did some electrochemical tests on this and found that it has a reversible redox reaction that occurs right at the potential where we operate our biocathode. And so we hypothesized that this is playing a role in improving our electron transport and improving cathode methane production, even without the addition of more zero-valent iron. On the right, you can see our results from our hydrogen sulfide tests with biogas in our biocathode. Surprisingly, to our surprise, we found that up to about 3% hydrogen sulfide in the biogas that comes into a biocathode, we can actually improve our methane output. We initially thought that it would be inhibitory. However, what we find is that the hydrogen sulfide is transported across the membrane from the anode, or from the cathode to the anode, where it is oxidized, improves the current output, and improves our methane output at the end. So based on our results, we have found that biocathode iron addition can significantly improve our energy recovery. Our hydrogen sulfide content, especially that we find in anaerobic digestion, can also improve our rate of biocathode energy recovery. And we know that the anaerobic digester biogas with this hydrogen sulfide in as a trace gas is a beneficial biocathode feedstock for biocathode energy recovery. And so this is a very positive result and suggests that our system will be able to do biogas upgrading very efficiently. However, the mechanisms behind some of these processes still need to be investigated, such as the role of iron and the role of sulfide in the effects that we see in the bioelectrochemical systems. But this has opened up a lot of new fascinating questions that are exciting and interdisciplinary, and we'll all work to develop a biocathode electrochemical system for resource recovery from wastewater. So what are our next steps? We are working to improve reactor architecture. So currently, many systems use the traditional H-style architecture, and this is not very efficient. So we're working to improve the reactor architecture, also investigate the mechanisms of electron transport. We are also trying to integrate nutrient recovery into our energy and wastewater treatment system. and then apply synthetic biology tools to our system to improve energy efficiency, and then finally scale up and pilot test these systems. So with that, I'd like to summarize and thank my team at San Diego State University, and point out that there's a lot of opportunity here for interdisciplinary collaboration in many different fields. And with that, I will turn it over to Christopher Heylman from Cal Poly San Luis Obispo, who will be talking about vascularized human tumors on a chip for drug screening.
All right, thank you very much, Christy. I'm very excited to be here this morning to share the work me and my students have been doing towards developing a vascularized human tumor on a chip. We're doing this for the purposes of screening novel pharmaceutical and drug compounds. So as we have a brief overview here, what we're attempting to do is build human tissues outside of the human body. And so to do this, we've developed a system that we term 3D microphysiological systems. So I'll kind of break that down. We're trying to do this in 3 dimensions because cells behave in a 3-dimensional construct much differently than they do in a 2D sheet. The majority of initial drug screen is done in 2D monolayer cultures. So we're trying to improve upon that to better mimic how these cells actually exist in the body and how they interact with drugs. And then the micro piece of that microphysiological is these systems are very small. So we can see that in this upper left here, you can see our actual device. It's the size of a microscope slide. It is a standard microscope slide. And then we put a small clear plastic polydimethylsiloxane PDMS device on top of that microscope slide. So in the center picture on the top row here, you can see our actual device. It's about the size of a dime. You can see we put some dye in it here to highlight the different inlets and outlets of the device. And then if we move across that top layer of images, you can see a micrograph of the actual chamber in which we grow these tissues. So the physiological piece of this microphysiological system is we introduce all of the biological components and attempt to recapitulate this tissue exactly as it exists in vivo. So some of those key components are a perfused microvasculature. So in the lower left schematic here, that's represented by these white squiggly lines. So it's essentially a capillary bed of blood vessels that would provide the nutrients and deliver the drugs to these tissues. We also have extracellular matrix. This is the proteins that serve as the scaffolding to hold the cells in place within this device and on which they build the tissue. We also have support cell populations, stromal cells. We use fibroblasts that help direct the formation of the tissue. And this device is a microfluidic device, so we're able to impart flow as well. And so we introduce flows that mimic the interstitial flows that are seen in native human tissues as well. Importantly, all the cells that we use in these devices are all human cells because we want to get those human-specific effects that really tell us how they're responding to these drugs in an accurate way. And so a typical drug development paradigm starts with exposing a drug candidate to human cells in a 2D monolayer in a dish. And so this gives us some information, but these cells will respond very differently when they're in the actual microenvironment of a tissue. So to get at that, traditionally, we then move into animal models to get these more tissue and organ level effects. And then subsequently, we go into human clinical trials, which is really the first time a drug candidate would see human cells in the actual microenvironment of a tissue or an organ. So what we're attempting to do with these devices is put that piece at the very front end of the drug development process. So we have more information about the likelihood of success once we've spent all the time and the resources to do those preclinical animal models and the really high cost and time associated with the clinical trials, most of which currently result in failure. So hopefully improve our odds of success once we get to clinical trials. So to achieve this, we've split this work up into 3 core projects in our lab, 3 silos, if you will. The first is focused on developing this microfluidic device itself, which is going to house the tissue. The second is this mix of all of the biological components, so the extracellular matrix proteins, the different cell types that go in there, the nutrient media that we provide them in order to facilitate the growth and development of these tissues. And then the last piece is really the functional use of this device, which is assessing how these tissues, once we've grown them, respond to drugs. So particularly, we're looking at tumors in this 3D microphysiological system approach. So we're growing tumors and then assessing how they respond to different drugs. So taking these 3 core projects one by one, the first one is the design of the microfluidic device itself. We do a pretty traditional approach to design here, design, build, test. So we start with making CAD drawings of the device itself. You can see one in the upper right here. Once we have the parameters worked out, we then do computational simulation of flow through this device. We use COMSOL multi-physics software to achieve this. Once we have a design we like, the simulations look good, then we actually go build the device. We have microfabrication facilities on campus here at Cal Poly. We use soft lithography techniques to develop the molds and then create our PDMS devices that we then bond to microscope slides to create the devices. Once we have the devices created, we then do physical validation of flow through the devices using fluorescent molecules and validating that the flow and velocities that we're getting through the device are similar to what we saw in our simulations. So here you can see some of the work on that physical flow validation. In the upper right here, we're using a fluorescent protein called Dykstra, and this is simulating loading through the device. So this would be when we're introducing all the biological components into the device. We want it to fill the chamber, as you see with the fluorescence filling the top of the major chamber there, but not leak out into the perfusion channels. So once we've introduced all that biological material, we then perfuse our nutrient media through the tissue, and then that would provide nutrients to the tissues as well as remove all the wastes as the tissues metabolize. To mimic that media perfusion, we use syringe pumps at defined flow rates to push 1 micron fluorescent beads through our devices, and then quantify the velocity of flow through the tissue compartment. And so we have some sample data here of that correlation of flow rate to velocities, and then match that up with our simulations to see if we're properly mimicking the native state in the body of the interstitial flows that we generally observe. Once we have the flow parameters down, we also need to ensure that we can properly sterilize these devices and culture cells within them. To validate this, we load different cell types into the cell, into the device, and then we perfuse with nutrient media for 24 hours and assess them for viability and retention. And so here we have our main cell types that we work with. The top row is 2 different types of fibroblasts. Those are those support cells that help facilitate the growth and development of the tissue. In the lower left are our endothelial cells that end up making the blood vessels. And then in the lower right, we have the current tumor cell line we're working with, which is a metastatic human colorectal cancer cell line. So moving on to the second core project, trying to optimize the combination of extracellular matrix proteins and different cell types within the device in order to facilitate the growth of these tissues. The way we do this is we use an extracellular matrix protein called fibrin that commonly appears in the body, but we start with its precursor, fibrinogen. And so we add the cells to fibrinogen, and right before we put them in the device, we add thrombin. This causes the fibrinogen to gel into fibrin. This is mimicking the coagulation cascade. This is the same way when you're bleeding that your body stops you from bleeding. So we're harnessing that biological reaction in order to capture the cells in a nice 3D distribution within the device and ensure that they don't flow out of the device once we start perfusing with the nutrient media. And so this is just kind of a sampling of the type of things we're working on in this space. On the right here, the 3 images across the top represent a mixture of endothelial cells that make up the blood vessels and those support fibroblast cells. You can see after 9 days of co-culturing these two cell types, we start to get nice vessel formation. Whereas in the lower row, if we're just looking at endothelial cells only, they tend to form more sheets or colonies of endothelial cells. So understanding the interaction of all these cell types is a prime activity within this silo. In the lower left, I have a sampling of what we're currently working on, where we've introduced our tumors into this mix of fibroblasts and endothelial cells as well. Now we have the tumor cells in green in the lower left and our endothelial cells that make up the blood vessels in red. Interesting finding here is that when we added our tumor cells to our mix of endothelial cells and fibroblasts that formerly formed vessels, with the tumor cells present, they no longer form vessels and we're starting to get sheets again. So one of our current challenges that we're trying to work through on the biology side. And then the last silo is assessing the response of these tissues to drugs. So we are using a colorectal cancer line. So one of the common treatments in the clinic of colorectal cancer is a chemotherapeutic called 5-fluorouracil. We culture our tumors in our extracellular matrix. We do confocal fluorescent microscopy. And then we get these images that you can see in the lower left. We have a grad student who worked on automating the image processing for this so that we start with this image. And then at the end, we can segment out the tumors and pull out a number of quantitative metrics from them, including size and shape parameters that help us assess the activity of the drug. So quickly looking at some data from that work, in the upper left here, you see average tumor size. There's two groupings of 3 bars here. The first grouping is a low density of tumor cells. The second is a high density. The first bar in each grouping is no drug, and then we have progressively more drug as we move from left to right. So with the low density, you can see that we get a nice reduction in tumor size as we increase the amount of drug. Same with the higher density, but we get a little less reduction in tumor size because we had more cells to start with. Similar patterns in the number of tumors. We have a nice drop off at a low density when we start adding drug. And when we have a higher density, we don't have much change at a low concentration of drug, but when we add more drug, we eventually are able to bring it down. So that kind of covers the technical context. I briefly wanted to touch on some lessons learned. I learned very quickly that in the CSU environment, it's hard to do things alone. So it really requires collaboration across campus, across departments, across the university. I've been able to source students from biomedical engineering, biology, chemistry, mechanical engineering. We require a lot of disciplines to pursue this type of research. Similarly, we require lots of expertise as well as instrumentation. So I've really been able to tap into all the resources that are available on campus. Similarly, this is not like your large research university type approach to research. You really have to break it down into bite-sized chunks, really small, achievable projects. So packaging them for master's theses, packaging them for summer research projects, that's been a new skill that I've had to develop in order to make this research successful. Similarly with funding, you don't have the big dollar amounts that you probably were used to at a large research university. So really piecing together lots of small amounts of funding has been kind of the name of the game. So summer research projects, we have a really nice regenerative medicine program here at Cal Poly, RSCA grants at the Cal Poly level, and then at the CSU level, I've been really involved with C-Superb in order to find collaborators and access funding there as well. So next steps in this line of research are to continue to optimize work within each of those silos, figure out the best flow parameters for the actual device, figure out the right combination of biological components, and then continue to improve our understanding of how these tissues respond to drugs. Ultimately, we want to bring this all together to have a vascularized tumor that we grow and develop within the chip that we can then introduce drugs to and assess their response. And then long term, we have partners in the pharmaceutical and biotech industry here in California that we hope to translate this work into their workflows to see if we can improve their drug discovery process. So I'm currently seeking collaborators in all the areas you see here in the red box. If you fall into one of these bins, please reach out. I'd love to talk with you more. I'm always looking for more people to help with us on this work. In summary, I just want to briefly reiterate that this is a complex project that we were able to achieve by breaking down into 3 smaller bins, access all the resources across campus to make it really a multidisciplinary effort. And I really think when this comes to fruition, it will be very impactful for the pharmaceutical and biotech industry here in California. So with that, I'd like to acknowledge all my grad students and undergrad students who do this work, as well as the funding sources that allow us to do this work. And with that, I'll look forward to answering questions in the Q&A session. And now I will turn it over to my colleague, Dr. Dahyun Oh at San Jose State University.
Thank you. Good morning, everyone. I have a little issue with my computer, but hopefully I stay very stable throughout my presentation. My name is Dahyun Oh. I'm an assistant professor from San Jose State University in the Department of Chemical and Materials Engineering. So here's my overview of presentation. So I'll introduce why I'm working on making a batteries, which is less flammable, I cannot really say non-flammable so far, but in the way that I'm trying to improve the safety of the battery is trying to focus on 2 different types of systems. First one is aqueous batteries, and the second one is a solid-state batteries. And I'll wrap up my presentation by sharing my future plan. So of course, the reason why I'm interested in my group of focusing on developing the safe battery is because you are carrying this device powered by batteries all the time. And now you are seeing even in the electric vehicle that it's powered by the battery. So it's of course like very critical to have a safe batteries. And the most flammable component inside the battery is the electrolyte. So people are trying to find the alternative electrolyte, such as aqueous, the water system, or the solid state electrolyte. But there are, of course, like pros and cons. For example, that for the water, it has a definitely lower electrochemical stability window, but so for the solid state, then we are still suffering from the low ionic conductivities and poor wettabilities of the electrolytes. And my contribution to the field in the aqueous battery was to experimentally detect how the water electrolysis is suppressed using the new type of aqueous electrolyte, which is also called the water in salt or the hydrate melt. How it looks like is in a conventional electrolyte, there are more water molecule that is surrounding the lithium ions, but in a super concentrated system, you basically have a less number of water molecule bound to the lithium salt. And there is a less available free water, so it's basically preventing the water to be electrolyzed in the battery cell. So on the right, it's a graph of the battery profile, the voltage profile, and the gas evolution data measured during the charging. On the top, it's a conventional electrolyte using the 1 molar electrolyte. Then you will see the hydrogen and oxygen evolution. But like on the bottom, once you switch to the concentrated electrolyte, then you are seeing the suppressed evolution of these gas molecules. So I'm keep working on this water in salt system. And the beauty of having this water in salt system is we can make the batteries, with some electrode that was not available in the previous aqueous system. So conventionally, the intrinsic stability window of water was around 1.23 volts, but with the water in salt system, you are able to use the electrode that can work up to like 3 volt differences. So in my lab, we're focusing on to use a TiO2 anode and the lithium manganese oxide cathode. And mainly my research group is focusing on how we can stabilize the surface of the anode TiO2 by introducing some of artificial SEI layers. So the first project that I want to introduce is the most recent publication from my research group by my master's student, Usha. She was a great student to work on this and wrap up the project and publish the paper before she graduated. And what we did is we introduced about like 5 nanometer thick carbon layer around the TiO2 nanoparticle and improved the cycle performance of the batteries by blocking the access of the water. So here you can see that at certain composition that we were able to achieve the higher cycle life of the batteries. And she also found that there are certain temperature ranges that she can apply to make a carbon layer, but which is relatively lower temperature, I would say, to pyrolyze the glucose. So the best performance that we had was achieved at around 600 degrees Celsius. So on the bottom, that you can see the capacity was way higher with the carbon layer that was annealed at 600 degrees Celsius. But 600 is still relatively lower to completely making a graphitic layer. So I would say this layer is still partially graphitic. But that's a good point because we don't want to completely conductive layer because it's going to be contributed like building super high SEI layer. So it should not conduct at all that much, but still ideally to blocking the access of the water molecule. So that was the first the project that we are focusing on. And we are also working on the solid state batteries. And this is a schematic figure from my other publication this year. It's a review paper talking about the challenges in the solid state batteries. And because the main point of having the solid state battery is to use the lithium metal as an anode, of course, there is always challenges in using the lithium metal, which is the dendrite formations or some chemical instability with the solid electrolyte. But what we are doing in our lab is focusing on the cathode side. And among many challenges, such as like poor contacts or forming some of the SEI layers or developing the space charge layer, we're focusing on how we can make a better contact between the solid electrolyte and the cathode side and improve the battery performance. So what we did basically in our lab is making a composite electrolyte using the polymer and the ceramic particle, that both of them are ionic conductors. And this project is done in collaboration with Professor John Lee in Mechanical Engineering Department. And this composite electrolyte is, we also determined the ionic conductivity by building the cell in-house. And this has about 10 to the negative 4.5 Siemens per centimeter ionic conductivity at room temperature, which is a very close number that is reported in the field. So with this solid electrolyte, what we made, we tested the full cell batteries, and seems like it's getting some capacity, but we're not fully happy with that. So what we are trying to do in this project recently is understand the impedance distribution inside the cell. So what we are trying is making a different combination of the cell components, such as like introducing some interlayer or making a composite electrode with an ionic conductor inside. So that's what we are trying and measuring the impedance and trying to decipher the impedance spectra and whether like each component, how it's contributing to overall cell performance. So that's an overview of what we are doing right now in my research lab. And my future plan for my research is definitely keep putting the effort on the super concentrated aqueous lithium-ion batteries. And in that, we wanted to mimic the composition of that is already composition of SEI layer is already observed in the super concentrated aqueous lithium-ion batteries, such as introducing some lithium fluoride component, which is less dissolvable in the water. So this requires an in situ surface analysis. So I'll look forward to hearing from you if you have the access to such an analysis technique. And also the other route that I'm taking is utilizing biomaterial to make a artificial SEI layer. So we're trying to understand how much of like conductivity of the carbon or how much of defective carbon are contributing to the battery performance. And for the solid state batteries, we want to expand to the synthesis of the inorganics. There are many interesting ionic conductors, but currently we are mixing the commercially available ionic conductors, but we want to make a step to synthesize these materials. So with that, I want to also thanks to all my students for their contribution and my collaborators at San Jose State, Professor Polarin and Professor Katie at San Jose State and Professor Minhan Lee at UC Merced, and the Berkeley Lab, a great staff member, Dr. Emery Chan and Rohan Daud, and thanks for all their help. And of course, I'd like to thank my founder, LG Chem, and Berkeley Lab and San Jose State University. So with that, thanks for your attention, and I would like to introduce my colleague, Subhradeep Roy.
Thank you, Dahyun. I'm a second-year assistant professor in the Department of Mechanical Engineering at California State University, Northridge. And my research area is in dynamical systems, and I'm very excited to talk about my research related to study of interactions in complex dynamical systems. So I would like to start with what are complex systems. The complex system, the main feature is that it contains a number of interacting units. And the interaction among these units lead to an emergent behavior that we see at the group level. Here are some examples of complex systems, for example, fish schools, bird flocks, where we see these beautiful patterns that emerge at the group level based on the interaction among these individuals. And these kind of systems I have interest in studying, and my approach has three main components. So first, I'm interested in studying interactions in real-world systems. And while studying these real-world systems, an important component is how we analyze this real-world dataset, for which I also develop data analytic tools. And then I apply these research findings that I learned from nature to design engineered systems. And in today's talk, I will try to give you a brief flavor of how I meet these three research objectives in my research. So I'll start with study of interactions in real-world systems. And the first example that I will present to you is interactions of flying bats. The question may arise, why bats? First of all, bats are social. They execute collective behavior. But unique to bats is that they use echolocation. And through echolocation, it's important because that promotes information sharing among the bats. So as an engineer, if we can learn how bats effectively use echolocation, we can build energy efficient drone swarms. So to understand how bats effectively use echolocation, we refine our research question and focus on a pair of bats. So specifically the research question here is when two bats fly together, one in the front, the other in the rear end, do they interact with each other or not? And in case they do, what is the directionality? Is the front bat influences the rear bat, or it's the other way around? So to answer these questions, here are the steps. So first we had to collect data of wild bats flying out of this cave, and this is our experimental site in China. And then we use this video data to extract the 3D coordinates to identify bats' flight trajectory. And then we analyze this data using different data analytic tools. So I'm going to skip the details and I'll present you the main research findings. So we used two different tools, and both of these tools detected there is a significant evidence of rear bat influencing the front bat. Now this is counterintuitive to us. This is because we use vision, and through vision we cannot take information from our rear end because that's our blind spot. But for the bats, it's possible. You can think of the rear bat makes a sound, and this blue region shows its acoustic beam. And if the front bat resides within this acoustic coverage, it can listen from the rear bat and then respond. So because of the sensing system they use, that gives the opportunity to have this rear to front coupling. Outside animal groups, I'm also interested in studying other real world systems. For example, study of interactions of neurons to study brain dynamics, for which I use EEG and fMRI data, and also how we humans interact to form opinions, for example, studying different public policies. So in each of these systems, I deal with some form of time series data, which brings the second component of my research, data analytic tools. So let's say we have two time series X and Y, and you can think of these two time series corresponds to bat's curvature data. So high value of curvature means the bat is taking a very short turn. Or you can think of these two time series corresponds to EEG data collected from two different EEG channels. So once we have these time series data, the question is how they are related. And there are three possibilities if they are related. For example, it may happen X influences Y, or it could be Y influencing X, or it could be a bidirectional influence where both X and Y have significant influence on each other. So to detect these relations among these time series variables, the tools that we use are called causal discovery tools. And these are the same set of tools I have used in my earlier studies. Transfer entropy, the top one, is basically an information theoretic approach, whereas convergent cross map, CCM, is built on dynamical systems theory. And since my postdoctoral studies, I've been developing our own tool based on a very different concept called dynamical symmetries. And these tools, I would like to point out that very important to learn the features of these real world interactions. The question is what we do after we learn these real world interactions. Next step automatically is to implement those into models for which I use two different analytical approach. The first one I use is using graph theory. So in this setup, interactions among these agents or individuals can be represented as graphs. For example, here is a schematic where you see these three robots, which are denoted by the nodes of the or the vertices of the graph, and the directed edges denote who they are interacting with. For example, a directed edge from 1 to 2 denotes that robot number 1 is taking information from 2. So once we model this interaction, next we can study different types of protocols. For example, a consensus protocol. Here is a time series plot of a consensus protocol where 6 agents start from different initial opinion, and then each time step they interact with its neighbors, they change their opinion through negotiation, and gradually they reach to a state where they share same agreement or reach to a point where they have same opinion. So this is also called a state of consensus, and these consensus protocols are often implemented on multi-robot teams where we want these robots to have aligned their motion. So I use these models to implement different bio-inspired interactions. For example, implementing a leader-follower interaction, for which I modify the property of the nodes of this graph, making some of the nodes as the leader nodes, and also implementing different types of interactions. For example, friendly and hostile interactions. Again, something we see in nature very commonly. So here we modify the property of these directed edges to build in this both friendly and hostile interaction. And then I use these models to study how fast the system will reach to convergence. So as an engineer, I care about not only the system reaching to convergence, but also about its speed. So I derive these closed form expressions of convergence speed in terms of system parameters. And I can come up with very counterintuitive results. For example, in this study, I found a counterintuitive result. For example, hostile interactions are enabling faster convergence for some system parameters. And another approach that I take is very different. It uses computer simulation technique. And in this simulation technique, one can simulate different collective behaviors that we see in nature. For example, fish schools, where each of these particles denotes each individual. And these yellow regions show their sensing region. For example, their field of vision. For example, the orange guy can only see the blue guy because the blue guy resides within this field of vision. And similarly, I can also implement different other types of sensing. For example, based on my earlier study with respect to bats, I incorporate an audition-based sensing, where instead of agents using vision, now they make sound. And these blue regions show their acoustic beams. And if an agent resides within this acoustic coverage, then only it can listen from its neighbors. So these two different sensing schemes differ in how the neighbors of an agent will be determined. And next I run simulations, and before I run simulation, I set certain rules how the agent's gonna modify their positions and directions at next time step based on these interactions. And in the simulations, I start with where all the agents are randomly oriented, and then after a certain time, I see in the visual mode multiple clusters evolve. And agents within these clusters are perfectly aligned, but these individual clusters move in random directions. Whereas in the audition mode, we see the entire group evolves as a single cluster and it's perfectly aligned. So this shows how sensing region has a big role to play in the group level behavior that emerges. So currently, after joining CSUN last year, I've been extending this research where I'm working with my graduate student, and we are implementing where the agents use both audition and visual sensing simultaneously. And also to a more realistic scenario, considering a three-dimensional collective behavior model. Another research direction that I have initiated since I joined CSUN is in the area of traffic and transportation. And this is based on my personal experience. Me moving from Blacksburg, Virginia, I found that traffic over here in Los Angeles is crazy. And so I thought I should study this system. And accordingly, here I'm interested in is how traffic congestion at one intersection influences the traffic congestion at the other intersection. And as many of you can guess, I'm using some kind of time series data. And here I'm using the flow rate. And this flow rate data is readily available from Caltransparent's website. Me and my graduate students are working on this and pre-processing the data so that we can start our analysis. And our aim is to build this influence network. The third topic is also related to traffic. Here, I'm more interested in the micro understanding of interactions among the car drivers. For example, a car driver, if we can quantify how much it interacts with its immediate front car and immediate rear car using information theory. So to summarize, I would say complex systems by nature requires a multidisciplinary approach, and successful completion requires collaboration. And previously, I have collaborated with researchers, and as a new faculty, I'm also looking forward to opportunities to collaborate. So if you are interested in any of these topics, I'm also interested in learning other research ongoing both at CSUN and other CSU campuses, if I can utilize my expertise to contribute in these ongoing projects. So here you can find more details. With that, I would like to thank you everyone for listening, and I will now hand it over to Dr. Gomez for the Q&A session.
Thank you, Subhradeep, and everybody for your really fine presentations. They were really great, and I'm sure the registrants were excited to hear all of them. We now have some time for some questions that were provided by the registrants when they registered several weeks ago. So the first question here is for Dr. Chris Bachman. Chris, as there are many reports of novel batteries in the news, what are some of the other technologies been worked on and that are upcoming?
Yeah, so I think most of the lithium-ion batteries that we're currently using are still based on technologies from the 1990s developed by Sony and John Goodenough, who actually last year got the Nobel Prize for that invention. And typically it'll be graphite on the negative electrode and lithium cobalt oxide on the positive, which is what they developed. And slowly, because of cobalt being expensive and toxic, they're replacing that with more and more nickel. So that's really the trend. The trend these days with batteries that are being used is to replace that cobalt with higher levels of nickels and manganese, and then also to implement more silicon into the graphite, so to increase the capacity of the cells. And so I think that's the trend. That's the trend in the near future. And then I think in the kind of upcoming 5 years, we'll see stuff like lithium metal being used with solid-state electrolytes, or other changes in the electrolyte. And then there's, I would say, in the 10 to 15-year period, there's kind of more far out changes that people are investigating, like replacing the positive electrode with an oxygen-based or a sulfur-based material, or to completely get rid of lithium-ion and begin to use something like sodium as a sodium battery as a less expensive material, especially where cost is a big driver. So,
okay,
yeah.
great. Thanks, Chris. We have another question here, this one for Chris Heylman. Chris, I guess it's a two-parter here. I'm going to add a little bit of my own interest also. Do you envision drugs being delivered via the blood vessel network in your chip systems? And based on my own long-term work in microfluidics, is there any interest in using non-PDMS-based platforms to also do comparable type of work?
Yeah, okay, good question. So I'll take the first question of are we intending to deliver the drugs via this vessel network? And the answer to that is yes. And that's easier said than done, but we have explored some strategies for interfacing the microfluidics of the chip, those channels where we can actually introduce nutrients and introduce drugs with that vessel network that we're growing within the central chamber. So really, that's where we want to end up, because that better mimics how the drugs are delivered in the human body, and we're absolutely moving in that direction. For the second part, we work with PDMS primarily just because of the ease of use, the ease of iterating quickly on our device designs. There has been some work in the area of paper microfluidics and the ability to immobilize different biological compounds, proteins, antibodies on paper microfluidics. We don't really work in that space. I have colleagues here at Cal Poly who do work on paper microfluidics, and they're using it primarily in the context of diagnostics. So introducing biological compounds of interest to assess what's present in a biofluid. But when it comes to cell culture, we have the additional requirements of having to be able to put this whole thing in an incubator, and the materials that we use to make our microfluidic devices survive that environment as well.
Great, thanks, Chris. Here's one for Christy now. What are some of the current research gaps that need to be filled in order to scale up bioelectrochemical systems for research recovery?
So we're actually very, very close on being able to scale up and pilot test energy recovery bioelectrochemical systems. I think a few of the gaps that we need to fill there is, number one, we need to look at the reactor architecture. So currently, a lot of systems use the traditional H-style architecture. However, that results in a lot of high overpotentials, and so in order to reduce that, we need to start looking at tubular systems, plate systems, comparing them and how they perform in a larger scale. So I think once we have kind of filled that gap, another area that really needs some research is in membrane science and materials science. So what we noticed is that the membrane materials really play a huge role in how the bioelectrochemical system operates, because the current membranes that we use, for example, Naphthalene-117, is very permeable to certain gases, and in particular CO2. However, it doesn't have as fast of a proton transport that we would like to see. So I think there's a lot of room for development there as well. On the nutrient recovery side, we have a lot more to go, I believe, before we start scaling those systems up. So currently, right now, our lab is working on proof of concept of developing a system that's integrated nutrient recovery and energy recovery with the wastewater treatment. So we're really kind of at the beginning stages of doing that, but I believe that there's great opportunity that we can jump forward with that relatively quickly based on our knowledge with the energy recovery systems and other nutrient recovery systems for electrochemical systems.
Great, thanks, Christy. Here's one for Sankha. Can perovskite materials be commercialized for applications as biomedical sensors?
Well, the answer is actually threefold, where we have to look into three different issues with perovskite materials. The first one being the toxicity of lead-based perovskites, which has the highest energy conversion efficiency. So we need to find materials or material systems or halides that can replace the lead-based perovskite materials. One area or multiple research areas has been looking into bismuth, tin, and even antimony, and even combination of one of those. So that's one thing that has to be looked at. The next thing is looking into the degradation characteristics. So these materials needs to be more stable. So that they're not stable. Even lead-based perovskites, they're using different methods, using scaffolding with polymers or copolymers, and trying to wrap these materials or devices up so that they don't degrade over time. The third aspect that needs to be looked into is if you are looking into non-toxic materials, you have to enhance or increase the energy conversion efficiency, as as I talked about. So these are the three areas that we need to improve for to actually commercialize perovskite materials as photoactive sensors or light-emitting devices for biomedical devices or biomedical wearable devices for diagnostics.
Great, thanks, Sankha. Here's one that a number of you might want to chime into and might take a while to answer. How do you run such productive research programs and balance your teaching responsibilities?
Can I start with that? I have been, in over the first few years, my wife was working in LA, so I had a lot of time on my hands, so that helped a lot for the first 3 years. Started 2015 here at Fresno State, so we did weekend commutes. It actually helped a lot because I used to stay in my office on the lab till like 11 p.m. at night. Uh, I was teaching 3 or 4 courses each semester, and then on the top of that, running a research lab where at least had 10 students, combination of graduate and undergraduate students. So it needs a lot of work initially, and I think as Chris Heylman mentioned, you need to find collaborators. So I had a lot of good collaborators. My postdoctoral advisor, he in Princeton Plasma Physics Lab. I found a few of my friends who joined different universities. Some of them are one universities, UC Merced, SUNY Buffalo, and whatnot, and also Portland State. So I started collaborating with them and actually distributed the workload with all of us. And when we apply for proposals, we actually do it together. I also found a lot of collaborators in Fresno State that helped me, helped all of us, get some funding. And that's my story. And there are different aspects, different ways to look at it. But I think most importantly, you have to spend a lot of time, and by juggling teaching and research and service activities, and it is difficult, but you can do it.
Yeah, and if I
Anyone
could
else?
chime in too, yeah. And so I think one thing that's been useful for me is taking my research students and kind of combining them into teams, with a graduate student with 1 or 2 undergraduates working with them. So in that regard, they're all working on the same thing. I can minimize the time that I have to sit down with them and train them on instrumentation and safety protocols. You can put that time investment in. And then as we're going through the research too, that helps because our weekly or biweekly check-in meetings, I'm meeting with 3 people at once rather than having to have individual meetings with all of them. So just trying to find little efficiencies like that where you can let them do independent work. They have a cohort that they can bounce questions off of before they come to you, and minimizing the amount of time that you need to be checking in on them, I think helps a lot.
Any other follow-up from anyone?
I think I could just say that I agree with what's been commented before. I think having teams of undergraduate students, the beginning is very difficult, just getting everyone trained. But I think if the students that are trained can then train the students that are coming into the lab, that really creates a system that then you can focus on guiding the research and not so much on the specific training that does happen at the beginning of the setting of a lab.
Okay. Well, I know when my lab was at its biggest, I had as many as 22, of which at one point I had 1 or 2 postdocs and about 5 master's students, and then a bunch of undergrads, and it was like kind of like having like a little one big army, but like little divisions of them. And so I would meet in the big group meeting, and then the sub group meetings, but I always found that it was good to pair off senior people with the younger people, because they were oftentimes better than I in terms of serving as mentors. And you kind of bring them up like a step ladder. You have the freshmen, and then the sophomores, and the juniors, and seniors, and the masters, and if you had a postdoc, it'd be great. And you just keep that ladder, keep those stairs going, and you always fill them and not have a break. If you have a break, you have some problems, but you always just have somebody in each one of those, multiple people in on each one of those stairs. So at least that's my take. So I'd like to certainly thank all of you for providing great insights on some of the questions, as well as the great research that you're doing. Speaker contact information is provided on let's see, I don't see the slide here, on one of the slides. For more information on any of the presentations, you of course can contact any of the speakers, as this might lead to a potential collaboration or proposal opportunity in the future. In addition, a link to the webcast will soon be made available on the STEMnet website. Answers to questions submitted during the reg period and not answered during the webcast will be provided on the open chat in the CSU STEMnet Facebook group page. In addition, we'll soon be sending a feedback survey to our registrants. Please take a moment to respond, as your feedback is important to us. For more information, please contact Monica Alacon or myself. We do have 3 scheduled webcasts coming up, as shown here. In October, we have the NSF Career Awardees. In November, Artificial Intelligence, Machine Learning. And then in December, we do have Exemplars in biology. So more information on that will be forthcoming. And finally, just a few closing remarks from me. The coronavirus pandemic is, and will continue to change higher education. Campuses are closed, online classes are the norm, and colleges announcing budget cuts and furloughs continue to grow. Students, faculty, staff, families, and others are wondering what lies for our futures. It is certainly a transformative time for higher education. Yet, let's look upon the present as an opportunity to prepare for tomorrow by examining innovative approaches to make substantive changes in education. Today is a unique time in all of our lives, and to make this change, not because we have to, but because it's the right thing to do. At the same time, as we rethink higher education, let's make sure we look at it through the lens of equity and opportunity for all. Let us look at these distributive alternative modalities, online learning, as a window to the future and to more engaging blended learning styles that draw on the best of both face-to-face and online learning. We have an opportunity to make better quality online learning tools, for as educators, we look to each other as trusted sources to help curate this information. Our collaboration should grow, thereby facilitating improved learning. Finally, the pandemic can bring us closer together, even in the face of physical distancing, words I prefer over social distancing, which I feel we should never really be. This is what the CSU is to me. It's why I chose to teach here for so many years, and now as executive director, be I think a bigger part of the CSU, and why I chose to graduate many, many years ago from a Cal State institution. So to all of you, may you be safe, be well, and have a great day. And I turn it over to Jennifer Wicks for a few closing words also.
Great. Thank you very much, Dr. Gomez. I just want to thank you very much for moderating, hosting this session, as you do with each of these in this series, and very exciting lineup to come, as you just mentioned, in October, November, and December. On the lower left of your screen, those are all listed. We do hope you all will join us. And again, I want to thank these wonderful exemplars in engineering and research for their presentations today and all their contributions. The recording will be sent out to all registrants, as noted there in the chat pod. We look forward to having you join us on the next session in October. Until then, have a wonderful Labor Day weekend, and we'll see you again very soon. Thank you.